Hello and very good evening to all of you. Welcome to this very special and exclusive episode of Eclectica, the Different News. Well, as you all know that I conduct this talk show quite often, and today I have my guest already inside the studio. Very good evening and very good afternoon to you, Dr. Tech. Very nice to have you, and thank you for accepting my invitation for coming over in Eclectica.
Thank you very much. It's been an honor to be joining here today. Thank you.
To all our audience, if you are here, just write in the comments, encourage and show your presence. That we also get motivated by that. Now you must be wondering that what this entire talk show would be about. Well, what we are going to address, or rather, what Dr. Tech will be telling us, is about a fascinating work that he has been doing for quite a few years now. And it is a technology which he has come up, which helps clinicians or which helps us to understand a malignant potential of a oral lesion. Dr. Tech, if I'm wrong, please correct me. So, you are very lucky. To all the audience, I'm telling you are very lucky to see and talk and interact with the inventor itself. And when you're here in the live, just make best use of this opportunity, because we will not get Dr. Tech every time like this on live. Okay, so Dr. Tech, uh, I would like to ask you that um, like you are a researcher, I know that well, in the field of molecular biology. Then my first question would be to you is that what motivated you to create the test, and how will this test improve oral cancer detection?
So we all know catching early the cancer is very important. So I believe that early cancer detection is the key to successful treatment. So if it is an early tumor, it's very small. So it's easier to treat, less harmful for the surgery. It's very simple. So you just remove it when it's very early. And we know that survival rate can increase significantly to over 80% if you treat early. So, what motivated me as a molecular biologist? I've been researching cancer genes using the technology PCR over 20 years now. And using PCR to detect genes, it's a very fast and simple technique. This makes me very angry. Why is it not widespread use for cancer detection? So that prompted me to investigate if I can use the technology and the genes that I discovered over the last 20 years to form a test that can actually quickly tell us whether a piece of biopsy is it high or low risk for cancer. And furthermore, we know that PCR is not a new technology. Actually, it's been invented in the 80s. So it's a well-established technology. And it's very unacceptable for patients to have to wait for up to 3 weeks for their pathology report. And when there is a fantastic technology there which can provide results within hours. So this prompted me to investigate, and I came up with this qMIDS technology, which is based on measuring 16 human genes using a tiny 1 millimeter, which is less than half a grain of rice size of tissue from the mouth. And we know that following the pandemic, the COVID pandemic, the PCR technology platform has been significantly upscaled globally to cope with viral testing demand. And this is an excellent opportunity to make use of this technology to detect cancer, I think. So, yeah, so
That's really fascinating to know the thought process which triggered you towards this wonderful work that you have been doing now. And we are very lucky to know directly from you. And well, to all our audience, I didn't formally introduce Dr. Tech, because if you go to cancer journal, you find qMIDS there. Am I right, Dr. Tech?
Yes, correct.
And just for your satisfaction, because I think Dr. Tech is everywhere now. So because qMIDS is something which is which is a very very important technology, and it is based on PCR, as Dr. Tech just now told us. While we go more forward in the conversation, uh, just to all of you, I just like to say that Dr. Tech is a senior lecturer in head and neck cancer, oral immunobiology and regenerative medicine, Barts and the London School of Medicine and Dentistry, Queen Mary University of London. I mean, that's a
Long.
name everybody loves to listen. Yeah. So, Dr. Tech, thank you for that wonderful answer. So, I was just wondering, Dr. Tech, that uh, you rightly said that PCR we know from long, and due to COVID, even common man now tells, okay, you have to do RT-PCR, whether they know or don't know, but it has become scaled up so much. You rightly said that. So, my next question is that you said that is the PCR based technology. So, what similarities does the test have compared with the PCR test for COVID-19 detection? If you can enlighten us on this.
So both tests are similar in a way that they both use RNA as their material for testing. So
Okay.
COVID uses RNA because the virus has got RNA in it, and the qMIDS detects the human RNA in the tissue. So both tests require a lab technician to run the PCR machine and to perform the analysis using a computer program. There are two differences. One of them is the PCR test for COVID requires a nasal or oral swab, as you know.
Right.
So you take the swab and then you put it in a bottle and you send it off to the lab. But for qMIDS, you need 1 millimeter biopsy tissue, and this has to be taken by either the dentist, oral surgeon, or the GP. So this is the difference in the method of collecting sample. And
Okay.
secondly, the difference between the COVID test and the qMIDS test is that COVID detects the viral RNA, which there are few genes only.
Mm.
But for qMIDS, we detect 16 human cancer genes. So that's the difference. We need more gene to validate a piece
Right.
of cancer, which is a very complex tissue. So
Correct.
that's the difference.
Okay, that's really amazing to know. So, Dr. Tech, I was just wondering that if you can tell us exactly how qMIDS, how exactly this qMIDS test work, and what kind of data will it be able to show us or demonstrate?
The unique feature of qMIDS is the 16 biomarkers that I've just said.
Right.
These markers were carefully selected over the last 15 years of my research in the lab. And these are actually unique genes that are upregulated in the head and neck cancer in the oral compartment
Mm.
for these particular type of cancer. So then the tissue biopsy is taken by the dentist. In the lab, the technician will digest the piece of tissue with an enzyme, and we add magnetic beads into the digested sample to extract RNA. So
Okay.
from the RNA, we mix it with PCR reagents and put it in the PCR machine, and the machine will measure the 16 biomarkers. And the biomarkers will be converted into a number that the computer program will calculate the cancer risk index.
Okay.
So the index, if it is above certain threshold, the piece of biopsy means that it has a high risk for cancer. And if it is below certain threshold, then it's normal. So given the qMIDS test is based on chemical detection,
Mm.
it can be automated. and quantified, the risk of human error in reporting the diagnostic tests is minimized.
I fall short of word after hearing all these things from you. I think it is a wonderful technology, and I think it is really going to change the way cancer is being diagnosed, particularly oral cancer when we talk about. And the clinician's pain is still remains, though we have very strong oral pathologists with us in our whole fraternity. But still, I think, as you said, that human error, it's always a problem. And another problem is that we at certain times, even with very experienced clinicians eyes, we cannot actually understand the malignant potential of a lesion. And I think qMIDS will actually help every clinician, I mean particularly dental clinician I'm talking about, in this respect. So Dr. Tech, so I am very interested to know that when will the test become available for use?
That's a very good question. Everyone asks me this question.
Yes.
So we have actually just published a paper validating the test on 535 patients from UK, India, and China, and we've shown
Okay.
very comparable results between the three populations. And this means that the test is very robust against differences in ethnicity and geography. And compared to histopathology, the qMIDS is over 90% effective in all the three countries patient tested. So the qMIDS test is now qualified for clinical study to confirm the diagnostic capability in the real life setting. And we need further funding, obviously, to carry this clinical prospective study so that it can be implemented to the healthcare system. So
Right.
before this is done, we will have to wait until we've done the clinical trial to
Correct.
validate the diagnostic capability in the real world setting.
Hmm.
So we are actually seeking clinicians for collaboration and investors, obviously, to fund the project and to help develop the test further. So for investors out there, this is a fantastic opportunity. So don't miss it.
Fantastic. Yes, absolutely. It's a very good opportunity for all scientists, clinicians, investors, because it's a very potential tool that you have developed. Well, we are almost at the concluding part of our session. So to all my audience, I would like to say that if you have any questions, please feel free to put it up in the comment box, so that we will have the privilege, and you will have the privilege, to get the answer from Dr. Tech himself. Because later on, if you ask the questions, maybe I just have to communicate with him and tell you the answers. But when Dr. Tech himself is here, it will be nice. If you have any questions, you can put it up. Well, so Dr. Tech, it's really fascinating to know about your work and about qMIDS in particular. So I'm sure that as a creative person, and a scientist's mind never stops. So so that way I would definitely I'm very very excited, or I'm very curious to know that what do you intend to work on, I mean work on next. What is your future plans?
Yeah. So at the moment, the qMIDS require, as we said already, one piece of biopsy tissue from the mouth.
Right.
And this is still an invasive procedure. So
Mm.
what I'm working now is to see whether brush biopsy means that a little brush that you
Yes.
brush against the lesion and collect some samples to see whether these kind of brushes can collect enough materials for the test to work. So it will be less invasive and less painful.
Mm.
And obviously, it will be simpler to collect by dentists or GPs to have the samples taken from the patient, or possibly in the future, the patients themselves could actually take using the
Correct.
brush biopsy. So to make it less invasive and more accessible to the wider population, especially to the rural area where the poor areas, um, where the disease is most prevalent.
Correct.
And we want to access to those population with a simple tool that can actually screen the samples, the population. So obviously this platform is not restricted to oral cancer, and
Okay.
it can be applied to other cancer types. So we are looking for new biomarkers for other
Mm.
disease, and we will use the same method and same algorithm that is behind this qMIDS that measures the gene expression to actually diagnose other diseases.
Mm.
So at the moment, we're trying to identify markers for head and neck cancer that are resistant to treatment. So we want to know which patient, when they have these biomarkers, and they would not be successful for treatment. For example, cisplatin resistance, and so certain drugs they may be resistant to it. But if we can know that person is resistant, then we wouldn't give them that particular drug. So we want to find out markers that are related and possibly use qPCR to test for that particular markers.
Thank you, Dr. Tech. It was wonderful talking to you, and I wish you all the best and all the best to qMIDS. And I hope very soon, with a lot of funding, and you can develop, and we clinicians can make best use of this qMIDS technology which you have invented. And thank you so much for being here. As of now, we don't have any questions, so we will bid goodbye, and I will keep meeting you, and I hope in the future also we conduct such programs where we will love to have you and your insights, and definitely all about qMIDS, because that's something a buzzword now. Thank you, Dr. Tech, for everything, and thank you everybody, and do subscribe my channel for watching more such interesting interviews. Thank you, Dr. Tech. Thank you so much.
Thank you, Dr. Gaji, for this opportunity. Thank you.
Thank you.
Bye bye.
Bye bye.